Welcome to everyone. It is a pleasure for me to welcome the speaker today, who is Petra Schwille. Petra is currently director at the Max Planck Institute in Munich, but is also honorary professor at the Ludwig Maximilian University in Munich. Petra started her brilliant career with the diploma in physics at Göttingen, but then switched her interest to very soon to biophysics, where she earned her PhD. Then she was a postdoc at Cornell until she returned back to Germany to establish her own group at the Max Planck Institute in Göttingen, where she's been leading the Department of Cellular and Molecular Biophysics until becoming the director of the present positions that she has now in Munich. Petra is an international recognized authority in the general field of artificial life and living technology, has been recognized by many prizes and recognitions, including the Leibniz Prize in 2011, the Bavarian Maximilian Order of Science and Arts in 2018, and the Karl Schweitzer Lecture Prize in 2020, and many others, of course. Petra is a member of fellows of many academies, including the EMBO, the Royal Microscopy Society and the Biophysics Society, and now, she's a fellow of ECLT, and we are very, happy to have her with us. And we are looking forward to your talk. So, Petra, please, without further ado, the floor is yours.
Thank you very much for inviting me both to your wonderful society team and also to give a talk. Yeah, so as was already said, in the last 20 years, we have been very much intrigued with the question whether there's something like a simple biology. And that's already an interesting question, probably, and a biologist would probably try to answer this question with no on the spot. And some biologists that I talk with are doing that all the time. But I would nevertheless like to motivate the question why we are actually interested in simple life at all. And that is a little bit, goes back in my own past, in my scientific, career development, when I started off doing single molecule method development, with very high precision. As you know, biophysical methods have now come down, especially with optical and fluorescence techniques, have come down to the level of single molecule. You can actually study single molecules. You can not only look at them by microscopy, you can also, do dynamic measurements on single molecules. The problem is the system that you're looking at should not be too complicated. And that's exactly the problem. If you take your single molecule measurements to cells, you're measuring all kinds of things, but you cannot really necessarily make sense of it. And that's the reason, because the cell is just super complicated. So if you want to, what you usually do in order to make a quantitative measurement, keep a lot of conditions the same and vary one, exactly that wouldn't work for cells. You cannot simply keep something the same and vary one parameters. This is something that is, to some extent, of course, you can do it in biology, but not to the molecular level where you're looking at single molecules, because you just simply don't even know how many molecular factors you have in. So we are faced with the enormous complexity. We are faced with, if you want to understand biology, we're faced with probably the most complex entity that you could study at all, with living systems. And of course, there is then the question, so what works so beautifully with physics, that you try to, to minimize your system of observation to a couple of, parameters, a couple of functions, a couple of modules, to make this, which has so beautifully been, accomplished with the development of physics for the hydrogen atom, right? Is there something like a hydrogen atom for biology? Something that we would say, well, this is actually the essence of a biological system. This is the smallest biological system that you could possibly study. So if you want to minimize your system, if you want to simplify your system, as physics does it for the hydrogen atom, or also chemistry, is there something like that in biology? And the answer is no, there's not. And the reason for that is that life has this ability to evolve. And presumably we don't know it because we cannot travel back in time and study life at the origin. Maybe in some future we will be able to study an earlier life form on a different planet, who knows that? But on Earth, it's too late, right? We are about 3.5 billions of years too late to understand how life looked at that time. But presumably it was very simple. It was much simpler, at least, than what we have now, because all that 3.5 billions of years, life has evolved. And like everything in evolution, it usually evolves to becoming a bit more complicated, right? So if you look the evolution of the car or the evolution of the TV or any technical element that we're looking at, evolution usually leads to more complexity, which makes it more robust and more efficient and more powerful. That's probably the same for life. But that does not really help us, right? So if we want to understand what would be, what are the ultimate modules that you would ask for in a system to call it alive, we just simply don't know. I mean, we can basically, of course, we can hold a couple of features that we know distinguishes life from anything else, such as self-replication, such as evolution, such as the ability to entertain itself, to metabolize, to take energy from the environment and sustain the energy metabolism all the time without running out. Information, having an information unit, having a self in terms of being a specific information unit that keeps itself alive or changes. All these features we know are typical for life. But how they did come how they came into existence, we don't know. And so the challenge is out there. How do we make simple life? And now we all know there are many ways, that we are not, our group is not the only one, and our approach is not the only one to arrive at a more minimal, more simplified life. Much more well-known is I would call it top-down. It's probably not quite correct, but the idea is that you take a living system and simplify the living system by taking, functions out of the living system, which, allows that thing to become simpler. That's what I would call the Craig Venter approach. Although he's not the only one of that approach, but probably the most successful, at least the loudest. And they have already come quite a long way. I mean, they have accomplished the reduction of a genome of a microorganism, mycoplasma, by half, which is amazing. From 1,000 genes, you can now go to 500 genes. And the thing is still kind of living, right? So it's not really happy, and it's not as successful as the nature they did. But it is, it's a synthetic organism where you basically arrived at from the top. Now, one problem with this approach, and that is very beautifully seen in Venter's results, is that of these 500 genes that are left in the organism that is, minimized, out of these 500 genes, about 150 or so are completely unknown in function. So we are having a lot of, black boxes in there, and that's naturally not what I like. I would like to have something that I understand, right? So the question is, knocking out things from the top might not really necessarily bring you back to the most simple. It might bring you in some sort of a local minimum from the top, which is not really giving you a good idea what the simplest system would be. And so if you really want to understand from first principles how living systems, are coming about, then you're basically left with the bottom-up approach, where you basically bring together parts or modules, which then, if you bring them together, develop into more than just the sum of their parts. And that's exactly the approach we are taking. But with a caveat. Of course, you know, if you really do it the clean way, you would have to bring together atoms, very simple molecules, molecules that were presumably there at the origin of life. This is something we cannot do. First of all, we don't exactly know which molecules were there at the origin of life. Also, there's, you know, some sort of converging evidence what that could have been. But we also know chemists, and we cannot, you know, these molecules are usually not some molecules that you can easily develop and engineer if you're not a super good chemist. So we are actually taking a shortcut. We are saying, well, we are taking functional molecules that are already out there, that are out there and in nature. And these functional molecules are proteins. And of course, now, you know, how can you try to assemble a minimal system if already start with proteins, which are supposedly very, very late products of evolution? Well, because only if you understand your parts, you can understand how the parts develop further, and you need some sort of a functionality to start with, otherwise you're not getting anywhere. And if we have something with proteins, we have functionality. We have a huge variety of functionalities. Some of them are even not fully understood. And as you may see in my talk, some of the functionalities only unfold if you take these proteins out of the cell and study them under completely different, similar, minimal conditions. So what we are trying to do in a, you know, systematic way is taking functional modules, bringing functional modules together and see how new functionality develops, and eventually the functionality that you're aiming at. And now the question is, what functionality are we aiming at? There are many, many different ones that you could aim at for a minimal system, for a minimal living system. Well, there we aim at, in our group, because we are membrane people, we aim at cell division. So we want to understand a minimal dividing, you know, system, something that has a membrane around and divides into. That's basically the technical goal of my group at the moment. Now we start with something, and that is something that we basically stumbled upon in the quest for looking at systems that that could be somehow initiating cell division. We stumbled over a very interesting molecular, you know, network, molecular system, and these are the so-called min proteins from E. coli. These proteins that you can see here are involved in cell division in bacteria, but they are interesting for us also in a different way because they are pattern forming. And if there's something important about a living system, then it's the ability to self-organize and form patterns. So life is all about self-organization. So meaning that you have molecules that use energy to, you know, self-organize in space and time. That's at the bottom of anything that is then in the end a living system. And these proteins that you can see here, they are doing that in a very exemplary way. So what you have there, I mean, the general idea is that you very clearly that a living system needs to do, it needs to unmix, right? So this is, you know, beating entropy in a way. Schrödinger has formulated that as neg entropy, so being able to create negative entropy to basically make a structure, make order into something on, you know, based on, you know, exploiting energy. So this is basically, and Turing has written this very beautifully in his seminal paper about morphogenesis. This is something that a living system can do. It can take energy, it can basically unmix molecules, it can beat entropy, it can create patterns and shapes and entertain that. Not only create it once, like in a crystal, so, but entertain it although all the molecules are exchanged. That is something special. And these proteins, the min proteins, which are involved in cell division in E. coli, they can do that. And what you see here are E. coli cells. They are rod-like bacteria. And these bacteria, if they want to divide, They basically assemble a division machinery in the middle, in the geometric middle, here along the long axis. And how do they assemble the division machinery? Well, they do that by oscillating other proteins back and forth. And these oscillations is something, gives something like a time signal, or if you will, a spatial-temporal signal to position the division axis into the middle. So the bacteria grow all the time, and only if they are long enough, the time between the two oscillations will be, right to make a division. And what looks so complicated in vivo, I mean, it looks simple, but you can imagine that it's complicated, but it's actually not. So what you have here are only two kinds of proteins. One is binding to the membrane, and the other one is taking it off the membrane. And that's actually all that you need in order to make such a pattern. The min proteins are operating in the way that the first one that binds the membrane is a ATPase, that is a soluble molecule. So upon ATP binding, it basically goes to the membrane. It changes its place in the cell. It's going from solution into the membrane, and then it cannot dissociate from the membrane, and it can also not hydrolyze ATP unless it brings, if you will, an antagonist of membrane binding or a catalyst of ATP hydrolysis, min E, to the very place where it is. And then only upon binding of this antagonist, the protein will be able to hydrolyze ATP and, dissociate again. And this whole process is non-linear. It's a cooperative process. So you need a couple of min proteins already on the membrane in order to recruit the antagonist. So it's not, a single molecule interacting with another single molecule. You need a certain concentration of membrane-bound min Ds in order to recruit the min E. And then also the turnover is a cooperative process. So they basically all dissociate at the same time. So it's hugely complicated. We still haven't figured out exactly how the mechanism works. It's certainly an allosteric transition going on, but for sure it yields this very beautiful spatial temporal patterns. So if you take the proteins out of the cell, purify them, bring them on a model membrane, you can actually appreciate that instead of having the oscillations, you have now traveling waves. Simply because there's no, it's not a boxed system, it's an open system, and these proteins don't have a reason to turn around. So only if you want to look at them, so what looks like a traveling wave is in fact nothing else than a cycle of binding and unbinding in a spatially coordinated fashion. So it's I like always to compare it with the Ola waves in soccer stadiums that run over the stadium and people don't move, right? They just stand up and sit down. And that's basically what we have here. Molecules coming out of solution, binding, and then dissociating again. So every single molecule has no impression of traveling. And this is the process that I explained to you. As I said, it's a cooperative process. So if you now want to understand what are the decisive factors of this process, well, obviously, a membrane affinity is a decisive parameter, and also ATP hydrolysis is a decisive parameter. And of course, we wanted to understand what of these parameters, how can we actually change the patterns? Can we change the dynamics? And this is actually possible with very, very subtle changes in the proteins. So if this is the wild type of the two molecules, and we get these patterns that I've already shown you and the oscillations back and forth, if you just change one amino acid in the antagonist in the min E membrane binding sequence, you can actually see that already the patterns look completely different and the pole-to-pole oscillation is lost. So you have a completely different dynamics. And if you take off the ability to bind the membranes in completion, so then or change this antagonist so that it cannot bind the membrane itself, then we still have patterns and we still have oscillations, but now it does something that would definitely not work in the cell. So what you can see here are I didn't show you that these are actually we locked these proteins up in a little bathtubs. So these are not cells, but these are bathtubs that are lined out with membrane like this here. It looks like a cell, it has the shape of a cell, but the proteins are it is just the proteins ATP and membrane in there. And if you see that here, then you can appreciate why this kind of a mutant would never work in a cell, right? It would not be able to position the division axis along the long axis. So this is something that's an experiment that you can only do in vitro. So intriguingly, if you go back to Turing and Turing's paper, it's never talked about moving patterns. It's always talked about static patterns. And the question is, can we also create static patterns with these proteins? And it appears that it's relatively easy to get static patterns that would then also be able to act as a spatial cue if you want to position something long term. Then it's only a couple of tweaks basically in the sequence of these proteins, so to make this still pattern forming, but to form static patterns. So we have in the last 20 years that we've been, almost 20 years that we've worked with this protein, we've now cloned so many different versions, and we believe that we understand what we have to do in order to make them do what we want to do. Something that was very interesting and puzzling for us, and here we come to the point of complexity versus simplicity. So this system is a little bit more complicated than I told you. So in principle, I told you that there's the MinD on the membrane, and it recruits the antagonist, and then the antagonist catalyzes the ATP hydrolysis, and both go back into solution. That's not entirely true. The antagonist needs to be switched into an open and active form in the presence of the membrane-bound MinD. So it's not sufficient to have it in solution and basically be recruited to the surface, but it has to be switched in an active form before it can catalyze the ATP hydrolysis and the unbinding to the membrane. And the question is why, right? Because we have a very good collaboration with the theory group, Erwin Frey here in Munich, and they have basically proposed to us that it should also work, this whole pattern forming process should also work if the molecule would not have to be switched on, but would be active all the time. And so we cloned it in a constitutively active form so that there was no switch. And we found that indeed you can see that the patterns are still formed with this simplified, non-switchable molecule. But the ability to form patterns is dramatically reduced in terms of the protein concentration range. So usually you need kind of a relatively similar concentration range for both proteins, the antagonist and MinD. And if you have the always active form, it's a very how would you say, strong switch from you see patterns to you see no patterns if you change the concentration of one of the molecules. So you can basically see, and this is the wild type where you have a relatively large window of opportunity, if you will, for a concentration balance between the two molecules. And I think this beautifully demonstrates that why biology is so complicated, because for biology the most important part is that things work, right? So you want to have resilience. You want a system that is not easily switched out of kicked out of balance. And this is much harder to do with the original wild type than with an engineered version that also works, but where it's just not resilient enough. So in an evolutionary game, this version here with the more complex, switchable antagonist would be, of course, superior. This would have the ability to, tolerate changes in concentration to a large extent, whereas this thing would just terminate to be able to divide. And I think that very nicely brings it before your eyes why biology is so complicated, because it needs to be resilient, it needs to survive. But that, of course, in the quest to understand principles doesn't really help, or that doesn't matter for us. We want to understand how patterns can be brought about in the first place, not how they are made stable or resilient. And for us, it was really interesting to see that you could cut off large portions of this antagonist and still keep the principal ability of pattern formation. So we saw that you can basically kill the membrane targeting that I showed you at the very beginning. Then we saw that you could also kill the conformational switch And then there's only one more feature besides the active catalytic helix of this molecule that makes it a bit complex, right? And that's a dimerization module. So in principle, it's a little helix, it's a little peptide helix that has 3 different motifs: a dimerization motif, a membrane targeting motif, and a switch. That's basically the design of this antagonist. And these 2 we have already found to be dispensable. We don't need them. So what about the dimerization? Well, the not so good news for the simplistic people is, well, one of the things you need, either you need a dimerization or you need the membrane binding. You cannot kick off both, but you can decide which one you take. You can have a dimerizer without the membrane binding, or you can have a membrane binding without a dimerizer. Now, this is the one. Or you can have, of course, both, as the wild type has it. But now the additional good news, besides being flexible, what kind of additional motif you put onto your catalytic helix, you don't have to take the native one. So we replaced both the membrane targeting sequence with a completely different one, and we also replaced the dimerizing motif with a completely different one. We even replaced it with DNA, as you can see here. So there's no protein at all. It's just a peptide helix with a DNA. And if you just have the complementary DNA strand on the other helix, then you can basically build an antagonist that is not a protein at all anymore. It just carries the functionality of proteins. Well, it has a peptide helix, that's for sure, but maybe even that could be replaced, because this peptide helix is, it's just an it's an activator for MinD. And who knows, maybe this could also be replaced by another chemical motif. So we have here now made the step away from the proteins. We have now basically understood what it takes to make patterns. It does not necessarily take a protein. It needs a dimerizing and a catalytic unit. And if you have the catalytic and the dimerizing unit, there you are with your, at least with the antagonist. We have not yet made, a non-protein version of the ATPase. That's of course a much harder thing to do, but it's not impossible, as we know. And maybe that's the future of a pattern-forming system, where you have both of the molecules actually completely synthetic. Okay, so I mean, this is good news for an approach where you want to simplify nature, not only having only 2 proteins, but also making them simpler than proteins. But something that we found very appealing and that we didn't expect at all is that these proteins, although they have a very clear function in the bacterial cell, and they are not present in any other cell, they are actually much more potent than we thought. And that's really something that is even better news, as I think. I mean, we completely accidentally found that. And what we looked at was basically what happens if you, of course, these patterns of the bacterial proteins, if you present them to eukaryotic people, they tell, well, that's a pattern of a bacterial protein. Why should I care? What can you do with such a pattern? Well, we found that these patterns of the bacterial proteins can interfere and can basically, act as a cue for other proteins that are not bacterial. We found that first with membrane-targeted M-cherry. M-cherry is just a fluorescent protein. Nobody cares about that protein, but it does not have any role in the bacterial cell. But nevertheless, we see that this protein is beautifully, positioned by our Min proteins, simply by, competing with the membrane binding. So this is a membrane-targeted M-cherry, as I said, and this is a membrane-targeted Min protein. And what you can see here is basically the membrane targeting of the Min protein is so much stronger that it kicks off the M-cherry and produces the reciprocal pattern. That's nice, but not super surprising, right? Of course, if you have competition reaction for a membrane, the stronger binder will outcompete the other one. And if you have a pattern in the one, you will have a pattern in the other one. It's not so surprising. It's beautiful that it looks like this. But what is surprising and what we did not expect, if you do not have a competition reaction for membrane binding, but if you have one binder in the membrane, which cannot dissociate. And there is another boring molecule. It's just a streptavidin, which we attach to the membrane with 2 membrane anchors. This thing can diffuse on the membrane, but it cannot dissociate. So what happens with that thing if min proteins are coming? Well, what happens, you can see here in this panel, the protein is pushed out of the way, not only, dissociated, as here in the M cherry, but really directionally transported. It's like a pump. What we have here is a pumping process, obviously energy driven because ATP is hydrolyzed. So we are pumping away this molecule here from the membrane. I mean, not from the membrane in total, but from a specific place in the membrane. So you're creating a pattern of this protein, and that could be any protein. The only condition is it needs to be membrane bound and it needs to diffuse. Nothing more needed for that. And then you can produce membrane gradients, which are at the bottom of nearly every biological important signal event, right? Wherever you have something going on that is interesting, you have a membrane gradient. It starts and then, not everything, but at least a lot of biological processes. So how do we rationalize that? Well, that was another very fruitful collaboration with our theory partners in Munich. The process is diffusophoresis, and it works like this, that you basically you have almost like a ratchet-like diffusion. So because these min proteins are so tightly membrane bound, they act as almost like an obstacle for the molecules in the membrane. So they can diffuse in one direction, but not in the other one, because there's the big fat min wave coming. So, and because of that, you can now actually use the molecular diffusion of these small blue molecules to basically push them in one direction. That's exactly what's happening here. And it depends, of course, on the diffusional mobility and also of the size of the molecules, right? The larger the interaction surface, the larger the hydrodynamic radius, if you will, the larger the effect will be on this membrane binder. And in order to show that this is true, we engineered membrane binding molecules, which are not proteins, but big fat origami molecules that are huge bricks, almost like bricks are made out of DNA, that we attach with membrane anchors to the membrane. And we have 2 different designs, one design with only a few anchors and another design with a lot of anchors. And what we see here is, first of all, we can this origami bricks can be transported by the min proteins. And they can even be sorted. They can be sorted according to the membrane footage of the binders. So these here, the bricks with the only few staples, and you always have to think that the min proteins, they're about in this height where the membrane binders are. So 2 membrane binders would interfere less with the min wave than a lot of membrane binders. So this thing will be much more efficiently transported by diffusophoresis, and that you can very beautifully see here in this superimposition of the 2 different colors. The green and the purple ones are basically the green one is the min protein, and the purple and the cyan are the different cargo molecules. So it means we cannot only transport molecules on the membrane by the min waves, we can also sort them. We can do a real diffusophoresis, right? A phoretic separation of different sizes of molecules to different spatial scales. Now, in the very end, I would like to show you another potential of the min proteins, which we did not expect, and we're very surprised to see and very much liked, because in the end, of course, what we wanted to do with these min proteins, we wanted to position a divisome machinery, and the divisome machinery in the bacterial cell is a huge, relatively complex protein system of different, about up to 20 proteins around one homologue of tubulin, FDSZ. FDSZ is basically almost like it's a structural protein, like a cytoskeleton of the bacterial cell. And that creates, together with many other proteins, a ring. And this ring will then be positioned by the min proteins, by the oscillation of the min proteins to the middle of the compartment. And so we wanted to, of course, to reconstitute a whole thing. But to get all of these in closed compartments, in closed membrane compartments, is a big challenge. But a couple of years ago, we accomplished the reconstitution of the min proteins, the oscillating min proteins in these compartments. These are giant unilamellar vesicles, which are basically our, most important candidates for minimal cell compartments. And here you can see what we have not yet accomplished is this elongation into a bacterial shape. In bacteria, you have a cell wall, which is basically making the shape of the bacteria in a vesicle without any shape-giving elements. You just have a sphere. And in a sphere, there's no pole, or at least no defined pole. So if you want to have a pole-to-pole oscillation, the proteins just don't know where the pole is. You get three different modes of dynamics. You get some sort of a pole-to-pole oscillation, which you can hopefully see in one of the, maybe in this here you can see here there's a pole-to-pole oscillation here. Some of them have, patterns where the molecules are going around in circles, and others, most of them actually have a complete volume to membrane oscillation, so that all the molecules go to the membrane, and in the other instance, all the molecules go to the lumen. And what we found is that if you now look at osmotically deflated vesicles, that we see basically a dramatic transformation of the shape of the compartment by this binding and dissociation, to the point that they are bouncing up and down. And if you have even more deflated vesicles, to the point that they're actually dividing in daughters, right? And this is only because you're playing by protein binding to the membrane. You're basically playing with membrane curvature. A binding from one side of the membrane is inducing a local curvature, and that basically results in a oscillation of membrane curvature, also surface tension, if you will. And that basically results in automatic, almost spontaneous division. It's not a full division to the point that the things are separated. That could be done, but it's relatively hard to accomplish that. But there is absolutely no structural protein in there. So what we originally wanted to have, a positioning of a structural ring-like shape by the min proteins, we seem not to need anymore, because the mins themselves are doing the job, right? They are basically transforming the membrane just because of the mechanical effect of binding a protein to the membrane. But of course, that's not eventually what we want. We want to have everything basically controllably with indeed mechanical element that induces the deformation right at the middle, not a ill-defined splitting. And so I just would like to show you the very end that we also are pretty close to that already. So this is the model that we have in mind. And this is basically the very recent work that a student and a postdoc of mine have accomplished, where we now indeed mix together everything, the structural protein that is supposed to make a ring, a contractile ring, and the min proteins in one vesicle. And you can see here very beautifully that exactly what is supposed to happen in a bacterial cell, namely the pole-to-pole oscillation driving together these filaments in the middle, right? So that you have a defined zones of division. So usually the structure, the filaments are everywhere, and only if the min proteins set up, they can actually initiate a real division ring. So far, no division here, because it's very, very hard to induce a force on a spherical vesicle. But what we found, if you just take all the native molecules in an in vitro expression, where you have much more than just just the proteins, the purified proteins, then you can actually beautifully see that does not only form a ring by itself, but the ring actually is able to induce a mechanical transformation. And that really beautifully shows now how it's driving it in a situation where you know, we are not yet there. That is a real division and a contraction of a ring to the scission of a compartment, but at least you can see that there's a transformation starting. And this is something we are very proud of. That's at the moment in revision. And let's hope that we get even further with, you know, making the real scission happen in such a minimized system. And with this, I would like to stop here and thank you for all your attention. Now I'm happy to take questions.
Thank you. Thank you, Petra. This is a virtual clap on behalf of the whole community.
Thank you.
I'm sure that there are, you know, I was fascinated, you know, and you know, I knew that it was going to be good, but the talk matches certainly my expectations. So I'm sure that there are questions. I think there is a question by Doron. No. Wait.
Yes, there is a question.
Please. Yes.
Thank you.
Please, Doron.
Petra, this was really fascinating.
Thank you.
But since I'm originally a chemical physicist, or physical chemist, but I did most of my life biology. Biology is full of these mechanisms. If you look at tubulin, if you look at muscle, if you look at GTP binding proteins for transduction and signal transduction and so on. So it appears as if this may have been a very early invention. I'm not sure I got, in terms of the biology of this particular bacterium, what it actually does and how similar it is to what we see as a very prevalent mechanism in which ATP or GTP supply energy for movement or for transduction, the association of and dissociation of proteins from each other and so on and so forth.
So this is what I showed you is basically all ATP driven. And the process with the bacteria, with this, the formation of these filaments is a GTP-driven formation.
So no, I wasn't so focusing on whether it is ATP or GTP, because for me it's the same. These two molecules are very similar.
Yeah.
So, but I'm asking about the principle of the functionality of utilizing the free energy of hydrolysis and then bringing it back.
That is, of course, the holy grail, really. I mean, in principle, I would say, whatever it is, if you have, in principle, what you need is an autocatalytic molecule that uses another chemical, whatever it is, as a fuel. And I mean, all of biology uses, you know, the usual suspects, right? So nucleotides, right? So that also, you know, NADH. So you have a couple of chemicals that, you know, have been, you know, established as the chemical fuels in our biological systems. But that doesn't necessarily mean that we need to have such molecules. I mean, the only thing that you need is you have to, you know, one autocatalytic reaction that basically uses another molecule as a fuel. I would love to build something on my own, but I'm not a chemist. I cannot do that. I mean, macromolecular chemistry can actually do quite a lot in that respect already. So making these, you know, almost metabolic machines, but you know, our ability to build something like that is limited. So we are basically using nature's one, you know, living systems, the usual suspects that I talked of. But of course, I mean, that would be something that would be the ultimate goal, to engineer a metabolism from bottom up.
Thank you.
Okay, thank you. Steen has a question, please.
Yeah, so it's a very nice talk. So I didn't quite, there was many details I'm sure I didn't really get. So which are these, you mentioned tubulins, what are these structures? You can see these, this band of things that are being, so to say, organized, I guess, by these mint proteins and
these are almost tubulins. Yeah, this is a bacterial tubulin. So it's a
bacterial tubulin. So exactly. You have the assembly of this primitive cytoskeletal elements that attaches to the, but will they always attach to the membrane? I thought
no, they will not. I was a bit short on that. So in the real cell, they have an adapter molecule. So in order to bind to the membrane, they have an adapter that brings it to the membrane. What we have done here is cheating a bit. Actually, in the very last movie, the adapter was in there. So there are basically 5 proteins in that we expressed in vitro. This is the adapter molecule, the tubulin homologue, and there are actually 3 min proteins. One of the min proteins I didn't talk about is basically just there to coordinate the FDSZ, the tubulin. But you can shortcut also the adapter protein by just attaching a membrane binding sequence to the molecule. So you can.
So, but how? I mean, the tubulin, they, as far as I remember, they don't have these contractive abilities. You need to have some kind of a what's it called, denine, or some muscle, or these molecules that can actually that has mobility. But or is it such that the tubulin molecules actually by themselves?
Exactly. That is the concept. So other than in a eukaryotic cell, where you have a template actin and a motor myosin or dynein or whatever, in the bacterial system, they have simplified, presumably, but it's not really understood, molecules that are called automotive. So they are doing some sort of a treadmilling, and by the treadmilling themselves initiate a force. But how they initiate a force and whether this force is somehow used for the division is not understood.
Okay, so it's actually thread. Okay, now I understand. So I can imagine anyway. So it's the thread milling, which we also have in regular microtubules. It's sort of linked to that. And if you then attach these tubes to the membrane, then you get I mean, you could certainly see that there was some
membrane
kind of mechanical.
Exactly.
Okay.
That's the concept. But as I said, it's not really understood. And some biologists would even deny that. They would say no. The only thing that the treadmilling is doing, it's templating the machinery to build the cell wall, and that machinery is then inducing the force to the membrane. So it's not only it cannot be done by these automotive filaments. But this, as I said, is debated at the moment. Our results point towards
it should be able
mechanical role, yes.
Because Hutani back in the early 90s, he was, I guess he was the first to show that if you have a self-assembly of microtubules inside a vesicle, you get this sort of American football shape thing, and these are the top tubulin molecules that are treadmilling in there, so they certainly can do it. But I don't know, these were from eukaryotic cells. So I don't know how it is with the. It's
actually very similar.
Okay.
So we believe that it can work, but how it can work, we have not yet understood.
Okay.
Okay, thank you. I see John has a question. Please, John.
Yeah, great talk, Petra. It's always fascinating to hear. So I have 2 questions. Maybe one question, which is the second question, will enable you to say a little bit about how you managed to see so clearly what you're seeing. What sort of fluorescence techniques you're using to make all of these images. But the first question is a sort of naive view of a simple way to do the cell division based on some of the mechanistic ingredients which you've uncovered. So I'm just going to play this very simple version back to you, and you can tell me why it's either too simplistic or whether that's the direction that one's going. Namely, we know from a lot of work on membrane protein segregation that you can create separated spatial patterns, stationary Turing patterns, and these are size dependent, so that if you're talking about a vesicle size, you can adjust things so you just have a pattern which has two components on the structure. So it may not be as focused as your ring, but you would have then a two-component pattern. And
yeah, I mean,
if you have, so the question is, if you have your mechanism of being able to control binding and positioning through your MIND proteins, why can't you use membrane curvature inducing proteins and the acquisition of these to just create the cell division process directly with the mechanisms that you have?
I mean, that is actually, in a way, that is what we try to do. I mean, membrane curvature inducing, I mean, first of all, you need something to, what you're saying, using, you know, unmixing of membrane components to position or to create patterns. That would be a static, in a way. It's a, you know, it's a condensation process. So you don't have, I mean, the beauty of our patterns is that they're dynamic. So molecules are exchanged all over the time, and that's exactly what you need. I mean, if you want to keep the system metabolically active, if you will, if you don't want to run into a energy minimum and stay there for the rest of your time, then you basically need that that continuous exchange, which you would not get with membrane, you know, elements. I mean, with lipids, you can, I mean, at least I'm not aware of a constant, you know, interchanging patterns. Usually if you have lipid phase separation or so, that's, you know, they separate and that's it basically. Then you have a pattern which will never change. And if you then have a division of your, I mean, that we've done in the past actually, where we have if you have different membrane domains like liquid ordered, liquid disordered, which is very beautifully unmixing, very beautiful patterns. But if you would have a division there, then you would end up with a you know, non-homogeneous.
Well, unless the divided system is sort of volume dependent, and so that you get triggered a new chemical reaction as a result of the division process.
But this would also be quite complicated. So I mean, unless you have some sort of other metabolic function that, you know, regenerates your molecules, which is probably also complicated to do, this would not really be, you know, it would not be able to cycle through many, you know, many versions of division. I mean, we have not solved the problem of division and regeneration at all, right? So if you want to have a real, you know, living mimicry, then the thing needs to regrow once it has divided. For that, you also need new material, and things become complicated there anyways, right? But at least the concept is that you have in principle, mixed open system that can go cyclic.
Okay, so thanks very much for helping me to catch up on that front. And the second question about the technique for measurement.
Well, there we have actually, that is really spectacularly simple. That's just confocal microscopy. And we are also doing turf microscopy. And of course, you can do all the kind of fancy stuff like high resolution. But really, it's not, I mean, that's actually the original goal of our minimizing biology, was that we can, you know, we can use our normal biophysical techniques to look at it.
Right. Nice.
Okay.
Good.
Thank you.
Thank you very much for the questions. Very nice questions.
Yes. I see. Are there any other questions for Petra? I see that you are even frying your team, so certainly on the theoretical side you are all set, I guess.
Yeah, well, I mean, we are happy to interact with anyone who wants to propose new, you know, models to us. There are still lots of loose ends to what we see here. Particularly,
but I was wondering how much theory can help in this such a, you know.
That's the point. I mean, I would think that where we are still, I mean, Erwin and his group, they are really fantastic in terms of, you know, understanding this pattern formation and all the self-organization, where we are a little bit still struggling, I have to say, understanding theoretical what's going on, are all these mechanical the mechanical aspects. Yes. What Steen asked about how do you actually engineer a membrane transforming force? It's not so clear.
I see. Yes. I see.
Excuse me. There is a question in the chat room.
Yes, Lucilla, maybe you want to ask. Unmute yourself.
She's saying that she has no voice because of COVID.
Okay.
So
maybe you can read the questions better.
I can read the question. I'm a cultural anthropologist. Really nice to be here. Thank you. And my question mainly referred to the research flow. First, I would like to know why choosing bacteria as a model organism for synthetic cell processes. Well, that's easy because bacteria are presumably simpler than eukaryotes. So if you want to have a simple one, you better start with a simple native one. And bacteria do not have a cell nucleus. They are just, in principle, they are a bag of proteins. Of course, they are not, but they're closer to the bag of proteins than a eukaryotic cell. And then the question is, I'm also curious to know a bit more about the disciplinary background of your team. Well, that's a very large range. We have physicists, chemists, biologists, even medical students. So basically everybody who is concerned with quantitative biology.
That's a perfect setup, yes. I think.
What expertise is lacking? Well, you know, it's not that we lack a specific expertise. I would say what we are lacking mostly are synthetic chemists. I mean, this is an art by itself, which we have never really, you know, mastered. And you know, the good thing or not so good thing about getting synthetic chemists, they have so many other things to do that give them more fame and money than what we
Okay, but I can tell that there are plenty of synthetic chemists here. So if you want to come, I'm sure they will welcome you.
I mean, if they find that interesting, they are very welcome.
Okay. Okay, so Petra, thank you very much. You know, so let's thank again.
Thank you. It was a great pleasure, and I really enjoyed your questions. Thank you.
Yes, thank you very much. Thank you for being with us, and hope to see you soon in person. And until that time,